Understanding evolutionary relationships is of interest for many biological disciplines. One way that we can infer evolutionary history is through sequencing and comparing parts of the genome across multiple species. In the past, traditional molecular approaches targeted very few well-known genes, which I refer to here as legacy genes. For many groups, this legacy data has accumulated across multiple studies, which often results in increased sampling from more closely related species. However, traditional sequencing techniques have been superseded by more cost-effective and modern techniques that can target thousands of genes from closely and distantly related species. Such techniques can involve sequence capture that target specific genomic regions, such as those that exhibit a high conservation across distantly related species, a characteristic that is not typical of most legacy genes. However, we often find gene sequences that were not intentionally targeted in our data set when using sequence capture, which I refer to as off-targets. Off-targets are typically ignored, yet they're highly abundant in these data sets and may prove useful when attempting to integrate legacy data and sequence capture data. Therefore, the primary objective of our study was to determine if we can recover legacy genes from the off-target sequences in an existing sequence capture data set comprised of 282 species of leaf-footed bugs. To do this, I targeted 15 mitochondrial and 7 nuclear legacy genes. To identify and extract mitochondrial legacy genes, I used the software MitoFinder, and I employed two different extraction approaches. For the first approach, I identified legacy genes through comparing them to the published mitogenomes of leaf-footed bugs. For the second approach, I then complemented those mitogenomes with individual gene sequences to see if I could increase recovery. To identify and extract nuclear legacy genes, I used NCBI to get gene sequences, and I then ran direct comparisons between these sequences and my sequence capture data set using BLAST tools. The results from this are as follows. The first graph is showing the average mitochondrial legacy gene recovery with standard error. On average, one more gene was recovered when complementing mitogenomes with individual gene sequences. The other two graphs showed the percentage of species in our sequence capture data set that had a particular mitochondrial or nuclear legacy gene recovered, and this varied across species per gene. Thus, the major findings from this project are as follows. Some legacy genes were successfully recovered from the off-target sequences in our sequence capture data set. Also, complementing mitogenomes with individual gene sequences increased recovery. And finally, variation seen in recovery of legacy genes may be due to the availability of reference sequences from more closely related species. Thus, given these findings, studies may integrate legacy data to create more complete phylogenomic data sets without increased cost. Additionally, future studies may look into how does the integration of this legacy data affect inferences for evolutionary history. Thank you.